DSP Cardiomyopathy. Now everybody heard the recording is in process, so welcome if you didn't hear me before. We're so happy that you've joined us for this really special update in research for Desmoplakin Cardiomyopathy with Dr. Helms and Dr. Smith. And just really briefly, I wanted to introduce myself to those of you I have not met in the group. I'm Christy Johnson, and I'm the founder and the admin of the DSP Cardiomyopathy support group. I started the group in 2021 after my family had significant issues with being diagnosed with Desmoplakin Cardiomyopathy in 2020. Anyone who wants to share their story, please reach out to me, and I'd love to share stories with you. But in the meantime, I just wanted to make sure that everybody knows about some of the things we do in this group. So we love to have doctors come and speak to us, and we've had some wonderful presentations and we'll continue to do so. We also have some informal chats where we get together in Zoom groups every month or every other month and just catch up. And then this is the place, of course, for you to talk, right? So write your questions, write your concerns in the Facebook group, and then take advantage of these very special times we have to talk to our doctors as well. So before I introduce our presenters today, I wanted to thank the DCM Foundation for supporting this webinar and ask you to check out the next Genetic Cardiomyopathy Awareness Consortium webinar. That's going to be about a brand new genetic testing program that's going to offer some free genetic tests as well as reduced costs depending on the situation. And you can find out more about that at geneticcardiomyopathy.org. So if you're interested, that webinar is taking place on May 28th. Ground rules for tonight. So everyone is muted for now. So please put your questions in the Q&A and I will facilitate a Q&A at the end, and Dr. Helms and Dr. Smith have saved quite a bit of time for Q&A. So you can think about your questions as they're speaking and put them in, or you can put them in at the end, either way. And this will last for about an hour, so we'll get to as many questions as we can within that time frame. But please remember not to ask personal medical questions because obviously, Dr. Smith and Dr. Helms don't know you. And even if they do, they can't answer personal medical questions tonight. With that, I'm going to head and introduce them to you, and then we're going to get started. So first of all, we have Dr. Helms, and he's the co-director of the Inherited Cardiomyopathy and Arrhythmia Clinic at the University of Michigan. In his clinical practice, he manages a variety of genetic heart conditions, including Desmoplakin Cardiomyopathy. He has worked with academic centers around the world to develop and expand research registries focused on these conditions. He also leads a translational science lab that is exploring novel treatments for genetic cardiomyopathy. Particular focus has been on Desmoplakin Cardiomyopathy. And then we have Dr. Eric Smith, and he's a clinical instructor in the Division of Cardiovascular Medicine and a key collaborator in the Inherited Cardiomyopathy program at the University of Michigan. His clinical expertise spans genetic cardiomyopathy, arrhythmia disorders, and pioneering gene therapy clinical trials. As a member of the Translational Science Lab led by Dr. Adam Helms, Dr. Smith dedicates much of his research to advancing the understanding and treatment of Desmoplakin Cardiomyopathy. His work aims to improve patient outcomes and drive innovative approaches to cardiovascular genetic diseases. So thank you both for joining us so much. I can't imagine having two bigger experts here with us tonight, and we're really excited for you to hear what you have to say. So with that, I'm going to turn it over to Dr. Helms and Dr. Smith.
Yeah, wonderful. Thanks so much for the kind introduction, Christy, and thank you very much for organizing this. It seems like just such a valuable resource for patients, and that you have been organizing these webinars is wonderful. So I'm going to go to share screen and we'll get started. Okay, you guys should be seeing my screen now.
Yes, looks great.
Okay, great. So today we're going to discuss a little bit of background on cardiomyopathy types, and then specifically how DSP cardiomyopathy is different, why it's different, how we try to understand that in general for heart muscle disorders in a research lab, and then how that can relate back to clinical research and clinical trials. And then I'll tell you a little bit about the trials that are immediately on the horizon and what we envision for the next 5 or 10 years. So just by way of background, genetic cardiomyopathies are heart muscle disorders. As this group is probably well aware, there's major types, including dilated, hypertrophic, or arrhythmogenic. The presentation for these conditions can range from childhood through late adulthood, even amongst members in the same family. The prevalence is 1 out of 250 in the population. So although desmoplakin cardiomyopathy might be a bit rare, the conditions as a whole are not that uncommon. So these are not conditions that people suffer with alone. There's a long pre-symptomatic phase. So typically, people don't really know that they have this condition for many years of life. And all of these conditions may lead to arrhythmias and potentially to heart failure, meaning that the heart fails to keep up with the demands of the body. So individuals become short of breath or develop congestive heart failure. Many genetic etiologies are now known. So that really puts us in an exciting position where we can start to develop treatments for these conditions. Desmoplakin cardiomyopathy is specifically caused by injury and scar in the heart muscle. So this is in contrast to the most common type of heart muscle dysfunction, where genetic variants really affect the contractile or squeezing function. So as distinguished from those genetic variants that affect the contractile apparatus of the heart, desmoplakin is a cell-to-cell linker protein. Patients often present with symptomatic muscle inflammation, or otherwise known as myocarditis. But most patients have these episodes without symptoms or come to attention because of palpitations, lightheadedness, fainting episodes, or shortness of breath. Over time, these areas of inflammation result in scar, and then that can worsen the overall squeezing function of the heart. And shown at the right is an image from an MRI where the bright white areas, so let me back up. This is the main pumping chamber of the heart, the left ventricle. The healthy muscle is dark gray, and these bright white areas are areas of injury or scar. And there's a particular pattern for desmoplakin cardiomyopathy where the scar is on the exterior muscle layer of the left ventricle. So individual heart muscle cells are held together very strongly at cell-to-cell junctions. These are called desmosomes. It's very different than skeletal muscle. So like your biceps, it just has a single muscle cell that goes the whole distance from the top to the bottom. And these muscle fibers and your skeletal muscle work together as individual units. Because the heart has a much more complicated shape, it requires there to be more of a network of muscle cells. And so those muscle cells in the network connect together through these very strong junctions. And the main component of those junctions that helps to glue these muscle cells together is the desmosome. You can kind of think of it like mortar and a brick wall. So desmoplakin, this is a cartoon of the molecules making up the desmosomes. So there's several different key components of the desmosomes. Desmoplakin is the main linker between those desmosomes and the muscle fibers. And so it's got a central area that's almost like a coiled spring, and then areas on either end that bind to the proteins at those locations. Patients with variants in these genes, typically the types of variants cause a loss of function. We've shown that from work in our group where we analyzed human heart tissue and identified a reduction in the desmoplakin gene and protein levels. So here in red, the desmoplakin is low compared to another type of heart muscle condition, and control in gray. So you can kind of think of it as that there's just not enough desmoplakin to serve its normal function. So the potential levels of treatment, we have come to think of this, of the process of developing desmoplakin cardiomyopathy as a sequential series of events. One is that first is that there's a loss of the desmoplakin function or amount of the protein. That then sensitizes the muscle to develop injury. That injury causes inflammation, and the inflammation over time leads to scar, a reduction in the heart function, and arrhythmias, irregular heart rhythms. Ideally, we would treat the root of this problem with gene therapy, but that hasn't been developed yet. Um, as a next option, we would think that the most attractive would be to prevent the injury in the first place. Another option would be to treat inflammation, which is important because inflammation can feed back and cause more muscle injury. And finally, there's treatments that improve chronic progression and symptoms. This is mostly what we have currently. So many of you may be on these medications, and they do help desmoplakin cardiomyopathy as they help other forms of heart muscle conditions. Inflammation is the closest to being available for routine treatment, and in the prime target where clinical trials have been developing towards so far. In our research lab, what we're primarily interested is these two boxes, and we'll get into that more. So we have different tools in the lab to try to understand conditions. We use two main, we call them model systems, but these would be a way to look at a condition without being able to study patients directly. So our two main model systems are, one is stem cell derived heart muscle cells. I'll explain that in detail. And the second is mouse models. I should give a disclaimer. We follow all ethical boards with all of their work in our lab. We'll tell you a little bit about cell work and what we do with mice in the lab, but everything is done in a humane and ethical standard. So for the cell work, we can either start with a skin biopsy from a patient or blood cells. And we can use a Nobel-winning technique. This is in 2012 Nobel Award, where scientists figured out how to reprogram those skin cells or blood cells to become a pluripotent stem cell-like cell type. This cell type can then be differentiated into other cells of a scientist's choice in a lab. And obviously, we make heart muscle cells. The technique takes about 2 weeks. It's actually pretty quick once somebody has stem cells banked. Creating a new stem cell line does take a little bit of effort. We have generated IPS, or for short for stem cell lines, both from patients, and we've also taken control lines, and we've used a technique called CRISPR editing, which you may have seen on the news. CRISPR editing literally allows us to manipulate DNA at any specific location we want to. We can introduce variants, we can introduce fluorescent proteins, all kinds of different techniques to understand the biology of a particular condition. So the stem cells they grow like weeds, and then we can differentiate them into heart muscle cells, and we can preserve them for various studies. This shows a typical differentiation of heart muscle cells. So they contract spontaneously and have all the major properties of regular heart muscle cells. So this gives us an infinite and ethical supply of cells to study the questions. Because the heart muscle cells in their native environment on a regular cell culture dish are chaotic and hard to study, we developed this technique where we generate tiny little heart muscle tissues on a elastomer silicone-like membrane. What's special about this is that it allows us to generate hundreds or thousands of these little tissues, all in the area that's about the size of a quarter. Each of these little tissues spontaneously contracts on its own, so we can measure its internal components with fluorescence techniques and high-powered microscopes. And we can also just measure its movement and calculate the contractile force generated by each of these tissues. All right, at this point, I'm going to turn it over to Eric. Eric Smith has been a wonderful colleague. He joined the group as he finished his cardiology fellowship several years ago. And I think really one of his main motivations has been to study this condition. He's put an enormous amount of work into developing the techniques you're going to see on the subsequent slides. And so I'll let Eric kick it off there.
Thanks, Adam. So I'm going to take you guys through some of the techniques that we use in lab, and a little bit about gene therapy. So what you see on the slide right now are those muscle bundles that Dr. Helms had previously showed, but they're tagged fluorescently with proteins. Certain proteins are tagged with a fluorescent marker so we can see them under the microscope. And so on the top, we've tagged part of the contractile apparatus of the cell with a red fluorescent protein. And the cells stay alive under the microscope and contract, and we can take videos of them. On the bottom, the protein that's tagged is actually desmoplakin. So desmoplakin links cells together. So where you see it on the screen there are the borders between the various cells within that microtissue, and they're glowing bright green because we're exciting the fluorescent part of the protein with a certain wavelength of light. You can go to the next slide, Adam. So we take these induced pluripotent stem cells that have been turned into cardiomyocytes and can use them to study the disease. But first, we need to make sure that they're accurately representing what we see in patients. And so Dr. Helms showed previously that the DSP protein level is reduced in patients that we see in clinic. And what this slide shows is that the cells that we take from those patients, either from skin biopsies or cells that we gene edit using CRISPR technology to reduce the desmoplakin protein, do indeed have low desmoplakin levels. So on the left, the black dots represent normal, healthy cells, and then the blue, purple, and red dots represent various disease lines that we use to study, all with lower amounts of desmoplakin protein. And we think that as the desmoplakin protein decreases, the severity of the disease increases. And we have some data to support that from our benchtop lab work that I'm going to show in the next couple slides here. So one of the primary questions about desmoplakin, what we asked initially, was that does the loss of this protein directly affect the way the heart muscle is contracting? And as Dr. Helms previously mentioned, this protein is known to be a cell-to-cell linker and not part of the contractile machinery that makes the cells get smaller and then bigger and smaller and then bigger. And so we can test this in the lab with the microbundles by using DSP patient cardiomyocytes and putting them into these rectangular tissues and measuring their contractility. And consistent with our hypothesis that desmoplakin does not affect contractility, we can show that the microbundles form normally. They create these rectangles as we would expect, and they contract normally. And when we compare their contractility to normal healthy cells, there's no difference. It's the same. And so that's what the graph that will appear here in a second shows. So all these black dots on here are healthy control cells, and the red dots are cardiomyocyte cells that came from patients. And they generally appear on the same spot on the graph there. So these rectangles that we image underneath the microscope are shrinking by about 5% each time they squeeze, and there's no difference between the desmoplakin patient lines and healthy control lines at baseline. So the next thing we sought to do was find a difference between the healthy cells and the desmoplakin deficient cells. And so we created a stress test, essentially, that we perform in the lab under the microscope using these patient-derived cell lines, and give the tissues a compound called endothelin 1, or ET1, that makes them contract harder. And so this is like an indirect way, or a way in the cells to increase the stress that they're experiencing and put them through a stress test. And this medicine, you could think of it as is mixed into the liquid that the cells are in and attaches to the cells and tells them to contract harder. The graph that's on the right, that I'll put up here in a second, shows that it does indeed work. And in both the control lines, the DSP plus plus and DSP trans minus minus, which is a disease line, the contractile velocity, the rate at which the cells are squeezing, increases similarly. So again, because desmoplakin doesn't directly affect contractility, that's something that we expected to see. The cells do respond to the medicine in the same way, but then things start diverging. So in the desmoplakin deficient cells, as we tell them to squeeze harder and stronger, they start pulling apart from each other. And the video on the top shows a tissue at baseline prior to the stress test. And our microscope setup in this technique allows us to go back to these same tiny tissues and see what happens after we do things to them. So the tissue on the bottom has been exposed to endothelin 1, and if you'll look in the center there, it's broken apart. And the endothelin 1 has caused that tissue to contract harder, and that part is normal. The contractility increases, the cells pull harder, but because the desmoplakin isn't anchoring the cells to each other as it should, the individual cardiomyocytes, the individual muscle cells rip apart from each other. And we think this is because of the lack of desmoplakin. You can go to the next one. So this is a time lapse of that same tissue that shows what's happening over time and that those cell junctions tearing apart. And what's really cool about this, and what's really really important about developing a test like this, is it allows us to now do interventions on these cells, give them medicines, provide them gene therapies, do different things to affect desmoplakin and see if we can rescue this this effect of the cells tearing apart from each other. We can go to the next slide. So our thought is that DSP loss of function variants are these mutations in the DSP gene that cause a reduced amount of the protein in the heart, sensitize the heart muscle to injury when there's excess stress around. Because desmoplakin isn't directly affecting contractility of the heart, it's not making the heart squeeze less strongly, it's just affecting the way the cells connect to each other. And as you stress those cells out more, they rip apart from each other, and then that sets off a whole cascade of events that are bad for the cells. You can go to the next slide. So how do we fix this? There's two different ways we're trying to go about this. We can restore the amount of desmoplakin protein in the cells with a gene-based therapy, or we can find a way to stabilize cell junctions without changing or doing anything to the actual desmoplakin gene. So when we think about gene therapies, I'm going to step back a little bit from desmoplakin specific therapies here and talk about what everyone is trying to do with gene therapies, because it's pretty confusing and there's a lot of stuff happening in the field right now. It's kind of exploding. So you'll see stuff out there about gene replacement therapy, and that's something that hypothetically could work for desmoplakin if you could take DNA or a new gene, put it into a virus, and get it into the heart, and the heart then expressed desmoplakin, you would fix that underlying problem, the low levels of desmoplakin. Unfortunately, the desmoplakin gene is too big, so it cannot fit into the current technology that we have. So we don't have a way to use gene replacement therapy to fix the desmoplakin problem. The next that you'll see around is something called gene silencing therapy, where we can use a technology to attach something to the DNA and affect the way in which the body generates the protein that comes from that DNA. And this is like the exact opposite of what we would want. The desmoplakin levels are already too low. We don't want to make them lower. So this technology has some applications, but not the right approach for desmoplakin. And then we could just directly edit the DNA. That technology exists. We can do that in the lab. That's how we created some of the desmoplakin cell lines that generated those videos you just saw. But it's very inefficient to get the gene editing machinery into the heart muscle. And the ways in which we may edit the gene are very complicated. So that this isn't currently a possibility for desmoplakin cardiomyopathy, but there are some diseases that are being targeted with this type of therapy. So you'll see out in the public right now gene replacement therapies for plakophilin 2 ARVC. There are clinical trials happening. Duchenne's muscular dystrophy, or DMD muscular dystrophy, actually has an approved therapy. Danon disease and hypertrophic cardiomyopathy all have gene replacement therapies available. Gene silencing therapy for something called TTR amyloid, where patients create too much of this TTR protein, and then direct gene editing for high cholesterol, for PCSK9, turning off that gene and then would lower people's cholesterol levels. And again, for TTR amyloid, altering that gene so it doesn't express anymore. But none of these are directly effective for desmoplakin cardiomyopathy. The other thing to be aware of and to understand about gene therapy is that we can primarily target the problem. So gene replacement therapy, in the case of that prior slide for, say, plakophilin 2 ARVC, is directly replacing the plakophilin 2 protein. So the problem is that plakophilin is low, and we're going to replace the amount of plakophilin with a gene delivered by a virus. It's the same idea that hypothetically you'd say, why can't we do that for desmoplakin? Primarily go after the problem, low desmoplakin levels. Let's fix the problem by increasing desmoplakin levels. But the gene is too big, desmoplakin's too big to do that. The other type of gene therapy you're going to start seeing is something called secondary targeting, where the loss of the gene or the protein, the mutation that a patient has, causes problems, and you may not be able to primarily affect that issue, but we find a secondary pathway, another protein that we could replace, or a different gene that we could edit that isn't directly the problem that has caused the disease, but indirectly helps fix either downstream effects of the disease or stabilize the protein in the heart to increase levels, but the effect is secondary instead of primary. So in our lab, we're able to rescue desmoplakin levels with a type of gene therapy that increases desmoplakin protein levels. You'll see in the picture on the screen under the untreated and treated column, if you look at the green dots on the left, there's less green dots than on the right. So we can drive up desmoplakin, which is labeled by those green dots. And when we do that, those previous cell ripping problems that we showed are fixed. So there are potential ways to increase desmoplakin protein levels, and it fixes the underlying problem. But this is still a long way away from clinic. But super encouraging that we can fix the problem that we see in the lab with our stress test.
Okay, thanks, Eric. So I'm going to wrap up here with a little section that we've done to develop a desmoplakin mouse, and then we'll go into an overview of the current clinical trials. Oops. Okay. So mouse models are essential for testing treatments prior to clinical trials. A lot of studies done in cells can be exciting and encouraging, but they require confirmation in the whole organ. Prior mouse model for DSP unfortunately doesn't, at least with single variants like patients have, it doesn't develop heart disease. So it's been published a little bit in the literature. We tried studying this model and really didn't get very far with it. And it's likely due to the fact that the mouse has a genetic resilience to loss of desmoplakin protein. So we measured this. Oh, it's like automatically going forward. Um, we we measured this, the protein levels over here at right, and see here with a single variant in desmoplakin, which is the same as the patient condition, the levels of protein only mildly drop compared to control. And then the mice don't develop any heart problems even out to fairly old age. So because of this, researchers have resorted to using a complete loss of desmoplakin in the heart. Desmoplakin is essential for other organs, skin and intestines included. So when one does this globally, the mouse wouldn't survive. But the trick is to express it to only cause loss of expression in the heart. And I think valuable insights have been learned from using this approach, but it does create such a drastic problem compared to the patient condition that it can lead to some limitations. And one is it could lead to incorrect development of treatments that are not relevant for patients, akin to like a false positive in the mouse studies, because things are so severe in this mouse model that maybe things that help it wouldn't help somebody with the patient condition. Or alternatively, they could lead to lack of benefit in patients because the model's too severe for some. Appears that there would be a lack of benefit because the model's too severe to really capture what might be beneficial in patients. So to get past this hurdle, we generated an approach based on human genetic insights, in which we have successfully now reduced levels of desmoplakin in a mouse model to match levels in patients. And the way we did this is we found the level at which protein levels would be, compensation would be overwhelmed in the mouse model. And then we further reduced by adding a second variant in desmoplakin that only caused a partial loss of function. And so we get a composite effect where the protein level comes down around to around 50%, which is exactly what we measure in patient heart samples. And then sure enough, this mouse model does exhibit in the heart areas of scar, just like we see in patients. And this is an example on the right where the scar area appears that bright red-green color. So taking all this together, our current approach is that we are using desmoplakin heart cell models to identify potential treatments. We then use the mouse model to test things that we can't understand just from looking at cells, things which I'm sure this group is interested in, like exercise, and to test treatment approaches to make sure that what we study in the cells is true to the whole organ. The mouse model's very new, so we haven't done extensive studies with it yet. We also use research registries. Many of you participate in these, and these are a very important resource to better understand a condition, how it progresses over time, and to better predict which individuals are going to suffer consequences. And this is critical, moreover, because of moving toward the clinic with more treatment options, I think, in the future. And then the final part is clinical trials. So clinical trials haven't quite gotten there yet for desmoplakin, but we'll talk about the options that are on the horizon. So we've really been primarily in this preclinical stage where we've been doing cell and animal studies, and not just us, other people are studying desmoplakin. And so this has led to some promising leads in the field. So far, and I would give a disclaimer that this may not be comprehensive because I may not know about things that are not publicly published publicly yet. But currently, what exists or what's out there that I know of is that there are 4 phase 1 studies. So phase 1 are safety studies. There's limited capacity to assess efficacy. They're not randomized, and they're generally pretty small populations. There are 4 studies that we're aware of, and one that's actively enrolling. Phase 2 would be after phase 1 has been approved by the FDA as showing a reasonable level of safety. And then it's allowed to go into phase 2 to not only further evaluate safety, but also to determine the effectiveness of the potential treatment. And there's one study that likely will begin enrolling within the next year that I'll tell you about. There are no phase 3 or 4. These would be graduation to hundreds or maybe even thousands of patients to evaluate, not for desmoplakin for phase 4 really, but to evaluate the treatment robustly and to evaluate long-term side effects. So I'll go through each of the potential trials that I'm aware of. This first one is an actively enrolling trial. It's studying antibody against CD14, which is a protein on certain types of immune cells that is part of the inflammation cascade. And so the company has developed an antibody that binds to this CD14 and prevents its activation, thereby downgrading inflammation. But it appears to be that the anti-CD14 is very well tolerated, and that it does not have the many of the immunosuppression complications of traditional agents like prednisone or many others. So the company's experience with some limited studies so far is that it does appear to be quite safe and well tolerated by patients. And it's being studied not just for arrhythmogenic cardiomyopathy, but for several other conditions with a component of inflammation as well. Currently, they are enrolling patients, but I'm only aware that one has been enrolled to date. I think the study's been kind of slow, in large part because it's only been a single center at Washington University, which is not necessarily a large referral center for arrhythmogenic cardiomyopathy. I don't know the logistic details beyond that. The study consists of sequential IV infusions of the IC14, and then individuals undergo blood tests and various diagnostic tests to evaluate the safety and potential efficacy following that. I do think that this study is likely to be extended possibly to Hopkins. I'm not sure if that is official yet or when enrollment may happen. This study could be considered by maybe by somebody who's had more issues with myocarditis. And I think that's kind of the goal of this study is to look at evidence of calming down the myocarditis. The next study is a FGF21 activator. FGF stands for fibroblast growth factor. This molecule in the body serves various roles, but it does partially control, regulate metabolism and inflammation. FGF21 has been actually quite extensively developed by industry with potential applications across a range of conditions. It actually leads to longer lived animals. So mice treated with FGF21 have a longer lifespan, and so it's had some attraction around that. It does seem that it is very safe to increase one's levels of FGF21. There's limited data on arrhythmogenic cardiomyopathy specifically. However, there has been a company that I'll get to more on the next slide that had developed this as a potential gene therapy strategy. As part of that, they do have preliminary data that suggests benefit in plakophilin version of arrhythmogenic cardiomyopathy. One nice thing about this particular study is that their particular compound, it's not a gene therapy, it's a small molecule approach. It's given as a antibody that triggers its response. So it's a once monthly injection, um, and not gene therapy. So it is shorter, easier logistically, and potentially safer than some other studies. The study, I think, will consist of serial blood test and a routine diagnostic monitoring, and it's probably going to be something in the order of 6 months, but the company is relatively new. It's called Preload Therapeutics, and I don't think they've announced their final plans. We are participating in a research project with Preload Therapeutics to evaluate specifically their approach in the cell models. Next is a FGF21 based gene therapy. This is developed by a company called Rejuvenate Bio. They're in California. It's following the same leads as what I described on the prior slide. Again, the safety profile should be quite good because FGF21 seems to be tolerated at higher levels in the body and maybe has some advantages. But this was now a gene therapy, so this does involve injection of adeno-associated virus. And one drawback of gene therapy at this point in time is that once one gets a single gene therapy treatment, you can't then be treated with another gene therapy because antibodies develop against the adeno-associated virus that would neutralize subsequent attempts at gene therapy. From what I can tell from the web, this company is still building up their preclinical data, and I do not see that they have announced an actual trial start date. And finally, there is another gene therapy strategy that is aimed toward increasing the levels of connexin 43. Connexin 43 is a protein in the heart that allows calcium to travel between heart muscle cells. It's part of what's important for heart muscle cells communicating with each other and triggering contraction. And connexin 43 level is known to be reduced in Desmoplakin Cardiomyopathy and profoundly in a mouse model knockout of Desmoplakin. In that model, gene therapy with connexin 43 restored much of the heart function and reduced arrhythmias. So it does seem promising from the complete knockout mouse model. I still do have some reservations for the reasons I mentioned earlier about how well those mouse models capture what's going on in patients. The study design has not been disclosed yet. Like the previous one, it would presumably be a single injection of an adeno-associated virus to deliver the connexin 43 gene therapy. And I also do not know the timeline. Their website also makes it appear as though they're still in the preclinical data, and I don't see any recent announcements. So where do these fit in the treatment, in the disease progression cascade we talked about earlier? IC14 is clearly targeting inflammation. And there could be then positive feedback toward reducing chronic muscle injury if inflammation is partially driving that. I do think this is a potentially promising strategy. FGF21, we think may improve inflammation based on some of the preclinical data from one of the companies. It could be that it helps to improve desmosome function, but I do think more work needs to be done for both of those treatments to understand fully what the mechanism of benefit would be. And finally, connexin 43, I think from the current information, we can conclude it does reduce arrhythmias. I'm less clear how much it directly would improve muscle function in the typical patient situation with a single loss of function genetic variant. Okay, so in summary, desmoplakin cardiomyopathy is due to a reduction of desmoplakin protein in the heart. This reduction causes extra stress at cell-cell junctions. Improved models are enabling us to test these treatments more accurately, including the heart cell stress test Eric showed you and this new mouse model. Um, Although standard primary gene therapy is not another approach, another genetic approach is able to improve levels of desmoplakin. And then we are also actively searching for other treatment options, some of which do appear to be promising leads, and we're following up on those. Uh, I want to thank foremost all the patients who've contributed to the research here, specifically to our registry, and providing for their enthusiasm for our group and clinic. Um, Also want to thank philanthropic support, both from the Cardiovascular Center at Michigan, but also patients. Have had several patients donate, including the Mouse Project got off the ground because of a very important patient donation. We have many members in the lab that have all contributed to this work, several collaborators in biomedical engineering, and a very vibrant clinical program that helps to support our work. So with that, I'll close and we can open up for questions.
Excellent. Thank you so much. That was so insightful. I know for so many of us that didn't understand all of that in the detail that you provided. And just thank you so much, because that was just a great overview. And so much to learn, right? So I'll be watching the recording again just to get the parts I didn't get. So thank you. So I do want to open it up for questions. And I had some that came in a little earlier. There's a couple people who couldn't be here. But I think I wanted to start, if it's okay, and then we'll get to, go ahead and put your questions in the Q&A. Sorry for anyone who does have questions, go ahead and throw them right in there and we'll get to them in a minute here. But one just sort of way to translate something that Dr. Smith said. So with the medicine that was used to inject the desmoplakin cells versus the regular, and understanding that the desmoplakin cells under stress started to, or the heart tissue started to split. I'm probably not saying that quite right, but started to become weaker. My question is can we translate that for patients to what could cause that stress on the heart? So we've heard, of course, about vigorous exercise, endurance exercise, potentially being a problem. I've also heard that major stressful issues, right, in normal life, just emotional stressful issues can cause the heart, of course, heart rate to increase. And therefore, is that something that we need to be considering? So I'm just wondering if you could tell me from a practical standpoint, how you as doctors may be able to help us translate that.
I mean, I think as scientists, we're going to help you translate that. And then
Okay.
as doctors, hopefully we can help guide people in clinic with what to do, because this is I mean, and this is what all the questions are going to be about tonight, I think, right? This is
Yeah.
like the most important. Well, A, how do I prevent this disease from progressing with a medicine or therapy? But B, like what can I do in my life to prevent the disease from progressing? And exercise is it's such an important aspect of everybody's life and the human body and health, that it's a really complicated answer, because it's not necessarily right to say exercise could be bad, therefore all exercise should be stopped, because that has potential detrimental effects too. There are lots of positive benefits to exercise in for the whole body. Um,
Mhm.
and I think one of the hopes from the cell-based stress test moving into the mouse is that we can, mice are active little creatures, and we can study them and see how the disease may develop and manifest itself in an animal model, to start understanding how it may affect patients in the clinic. And I think we don't have that data yet, so it's hard to say. And the clinical data gathering data from these registries and stuff sometimes can help answer these questions, and it has in diseases like plakophilin 2, ARVC, where there's very clearly a link to exercise and endurance and endurance exercise and the quantity of it and it leading to progression of disease, and the entity of like endurance exercise ARVC that may be gene elusive, but patients develop the disease who have exercised at very high levels. That link is less clear in Desmoplakin cardiomyopathy. Um, it hasn't come out of the patient registries that we have as clearly as it has for other types of genetic.
Right. I mean, there was, not to interrupt you,
Cardiomyopathy.
Dr. Smith, but there was the recent Johns Hopkins DSP related exercise study that was released with, and I don't know if you all have something to say about that, but it did show that they believed based on that research study that there is a link to endurance exercise with this DSP, which you said initially. So
I mean, I think there probably is the like physiologically, I mean, I think there is probably a link between stress, um, exercise, and progression of disease, as the cell model shows. But the what's safe and the degree of of exercise that one can participate in that's going to bring them benefits of exercise, but prevent their disease from progressing substantially is a really hard, um,
yes, and
is a
that's the
real,
question everyone has.
yeah, is a
They want
really
to know what they
hard,
can do and what they can't.
right.
Yeah.
I don't
Yep.
know, Adam, what do you think?
Yeah, Dr. Helms.
Yeah. I mean, I can comment on the paper from the Hopkins group. I think it's a nice study, but it's not totally conclusive.
Mhm.
It's not like PKP2. So in PKP2, it's just very clear cut. Heavy levels of exercise definitely contribute to the developing the condition and definitely contribute to progression. It's very clear cut from the literature. In the paper on desmoplakin from the Hopkins group, they did not find a link to between exercise and adverse events from heart failure or arrhythmias. But it was a bit, it's a bit complicated because they also have been in the mindset of telling people to mostly stop exercise at the first clinic visit. So it then becomes a confounded is the word. Like the fact that they're intervening at the first visit with this advice then sort of changes the potential history. And then because of the fact that I don't think the relationship is as strong for desmoplakin as PKP2, then you could miss signal from this. But what was clearly shown is that myocarditis episodes were more common.
Mhm.
And I think that does fit with our understanding of the biology of the condition. So we had already been giving patients guidance to stick to more moderate levels of exercise. This
Mhm.
is such an individualized decision, and some people, a lot of activity is normal and they're used to it. Maybe it's less stress, and somebody else, it could be, quite sedentary, and it's a very different discussion. So I wouldn't say that we have a one size fits all answer, but yes, I think we do think that intense, very high levels of exercise, very high volumes of endurance exercise probably do contribute to progression. And that's generally our advice in our clinic is to not engage in those intense levels of exercise if people have evidence of problems on their diagnostic scans. So not
Okay.
necessarily somebody who's never shown signs of desmoplakin cardiomyopathy, like a gene positive carrier, but somebody who's has the condition, has abnormalities on their test. Our general advice is to not engage in highly intense activities.
Okay, so that you just hit on these next two questions. So
Yeah,
the
I was
first,
reading through the chat, so I was trying to
so
get a
you can
couple other ones
see,
there.
so there is that person who is saying, is it a risk factor for developing the disease? So is intense exercise or endurance exercise a risk? If you're gene positive, is it a risk for developing the disease?
I'd say our level of evidence is not sufficient enough to answer that question
Okay.
definitively. Our hunch would be that it could be one contributing factor, but there is also obviously a lot of person-to-person variability.
Yep.
So one person's condition may be quite severe no matter what they do, and another may be able to do these higher intensity activities and be okay.
Hmm.
And that variability, currently, we can't predict it. It's
Okay.
just something that's part of the condition and human variation.
And then, so that next one, so what about your advice for children, especially young children? So what would you advise for a gene-specific child with no, no signs of disease? What can they and can't they do? How do you help
Well,
people?
I think what I typically will do is um, it's really important, I think, for people to understand what their kid's genotype may be. And
Mm-hmm.
then because I think there's a good chance that down the road we get more definitive evidence that very high level competitive sports are not going to be good in this disease.
Mm-hmm.
Um, to not necessarily like again make that kid a couch potato, but as they grow up through life, direct them towards activities that are not becoming an elite level athlete. And so I think you have the opportunity to do that when kids are young rather than waiting or getting them enrolled in travel soccer and they get a Division I scholarship.
Mm-hmm.
Now their MRI is starting to show fibrosis and evidence of
Mm-hmm.
disease, and you're stuck like in a situation that's really unfortunate because it may be unsafe for them to continue. And so what
Mm-hmm.
I typically tell patients with young kids is you don't need to like keep them from running around on the playground. They can participate in normal athletics, but as they start moving into the more competitive, very high level sports that a lot of kids are participating now as teenagers, um, guide them away from that at an early stage, so you don't have to worry about it.
Mm-hmm. And I
And
mean,
of course,
personal,
shared decision making. And then if they do choose to participate, then the annual screening is super important. Because then if you find abnormalities, then you have a chance to redirect.
Yeah,
yep.
I mean that's we can watch things super close, and I think it's important to find a doctor that's willing to to go through the shared decision making process with you and work with patients on an individual level to figure out what's best. Because some families and patients may decide that what's best for them is to remain a high level soccer player, but with the understanding that the disease could progress. And so we're going to watch it really closely as you do that and have a plan to stop or cut back if we start to see problems occur, as opposed to saying like, absolutely not, you can't do this, like either stop doing it or stop seeing me sort of thing.
Yep. Yes, that's great. Thank you so much. And I know we only have a couple minutes left and we've got quite a few questions coming in. Do you have a
Can
hard
I
stop?
choose some to answer? Okay.
Well, yes, I'd love
I
if you could
can.
address the disease progression because I get that a lot in the group too. So once you see
Oh, well, let me
symptoms
scroll
and
up. Which one's
stabilize,
about?
or is it a downhill from there? So do people stabilize over time sometimes, or is it
Yeah,
downhill?
I think, that is very different from person to person. So the individuals we are most concerned about are those who develop problems very early in life, and particularly with recurrent episodes of myocarditis. Actually, a question just popped up about that. Yes, there is a link between, if not even super clearly in the literature, but in our experience, between higher frequency of hot phases and disease progression. And we've started to treat those more assertively using immunosuppression. I
I
think that
think
was
so.
another question somebody had.
Yeah, Andrea asked a question about mycophenolate and colchicine, and Dr. Helms and I have been starting to use immunosuppressive therapy in some patients who have very frequent hot phase episodes to try to calm some of that down. And there's a little bit of data out of the registry we participate with Hopkins that supports that thought process a little bit. But I think we also feel like we're kind of treating this with a sledgehammer. And so that's why we're trying to find more targeted immunosuppressive therapies. But there is a role, I think, in patients who have very frequent hot phase episodes.
All right, Dr. Helms, I'm going to let you read just so I don't, so we got.
Oh, yeah. Okay.
More
Well,
minutes to address
I
the
like
ones
this
you want to.
question about routinely testing the DSP levels. We would love to do that. The problem is most people don't want to sign up for voluntary heart biopsies because they're not without risk. So we don't really have a way, unless we make iPS cell cardiomyocytes from every person, but those are quite expensive and individually time consuming. Maybe one day if that becomes more efficient.
And that was a question because they all can't see them. So that was just a question about whether we can routinely test the DSP levels. So that was the answer to that. And that was a great answer.
There's a question of testing troponin levels after exercise, which is an interesting question, especially with the troponin tests we have now. They're ultra sensitive. It sounds like this was a particular patient that was testing themselves to see how he was doing. I mean, I don't, that's a potentially interesting scientific question. If you had patients that were exercising otherwise normally to check high sensitivity troponin levels in them after significant exercise. And I mean, if you were to see elevations in those post exercise, that would be concerning to me. That there is low level damage happening because you shouldn't elevate your troponin levels just from exercise.
Great. Thank you. There's another exercise. Oh, really quickly, do you see anything different in the right-sided versus left-sided DSP in terms of exercise or any other components of the disease that you want to comment on?
Right. Pure isolated right-sided DSP is extremely rare. So it's only about 1% or 2% of very large cohorts. So
Wow.
most patients with right-sided problems have both of the chambers involved. There are a couple out there, but it's quite rare. We just looked at this again in a large multi-center cohort with the Hopkins group and 40 others around the world. It was about 1% right-sided. So the number is so small. In theory, it would make maybe more sense because it would be more similar to PKP2 at that point, but the numbers are too small to really make a conclusion.
There's a question about are PVCs related directly to the progression of DSP? If the PVCs are almost non-existent, does that mean the condition could be in remission? We think PVCs are pretty important in DSP cardiomyopathy, and their presence being an important component of diagnostic criteria and also something that we use in clinic to assess disease progression if PVCs start to increase. I think it's fairly uncommon for a patient to have significant PVCs and then them all completely disappear. Though there are patients that may have evidence of disease in other ways and have less PVCs or have a low amount of PVCs, and that seems to be a good thing. It's PVCs roll into the risk assessment for ventricular arrhythmias, and a patient, two equal patients, the one with PVCs, the one without, I would consider the presence of PVCs suggesting more severe disease. But we don't see them come and go completely.
Thank you. That's great. The other one, one more, and then I would let you guys go, and this is one that we get a lot as well, is diet. Is there anything that you've discovered when it comes to people's diets that make a difference in lifestyle for DSP cardiomyopathy besides obviously being healthy? Someone here is talking about inflammation in the body overall and how higher levels of protein, lower carbohydrates, she feels have helped her. So just as a overall consideration to diet, and the answer maybe you don't know, but if there is any research or anything that you can share, that's a huge question that comes up all the time.
I think the diet that's described here is great. So I would encourage continuing, a low carb, um, high fiber diet. That's a general heart healthy diet and good for your body overall. I do think it's possible that having a lot of processed sugar and fat in the diet could create a inflammatory state, and in theory, maybe that could contribute to the cardiomyopathy, but we wouldn't have any definitive evidence to be able to say that for sure. But I think the healthy diet is great overall and maybe good for the cardiomyopathy.
I will say anecdotally, many people have told me that carbs and simple sugars make their PVCs worse, which is like
Yeah,
kind
people
of,
have told me that too. Yeah.
yeah, but we don't have any like definitive proof of that.
Great. I think you hit on a lot of this. I'm looking at one. Can everyday mild, moderate physical and mental stress cause muscle injury? I don't know if you know the answer to that, but if you do, we'd love to hear it.
So we're not actually totally sure what triggers these myocarditis episodes in patients. We always get a detailed history, and it's usually not that they've been previously exercising intensively, because we always ask about that. Um, I have had a couple people clearly describe that like recurrent episodes that were after periods of high stress in their life, like pulling all-nighters or some kind of just stressful thing in their life. And so I'd like to think that's some true association, but exactly how that connects to heart function, I'm not totally sure.
Okay, that's great. Well, I know we've kept you a few minutes over, so thank you so much. I don't know if there was any final words that you wanted to say before we go tonight.
This was great. Thanks so much, Christy, for organizing this and for all of you guys bringing your questions, and really our pleasure to talk with you all today.
Yes, and let us know too if there's ways we can continue to support your research as a patient group. I know this patient group is very active, so please, if there's anything that we can put out in our Facebook group to support you or to, we will. So
We'll
we'd love,
certainly support the NIH, and
yes,
this stuff is in trouble these days, but the NIH does mostly 98% good work, and we're NIH funded, and hopefully that continues. Without the NIH, there would really be no academic universities, regardless of how much philanthropy or other kinds of support people could get. So, yeah, be active on the behalf of health research in your communities.
We're
great.
we're indirectly thinking about all of you every day because we spend all of our time studying this disease and working on it. So we may
And we
not be
so
able to,
appreciate it.
we may
We
not be
so
able to see you
appreciate it.
in we may not be able to see you in clinic, but like uh we're working on the stuff in the lab, making the cool little cell-based stress tests, doing things to try to figure this out because there's a lot of unanswered questions, but there's also a lot of potential exciting stuff coming down the road. So
Excellent. Well, I cannot thank you enough for your time and your expertise that you've shared. And I'm volunteering to be a guinea pig anytime. Give me a call. I bet you'll have lots of other people as well. But thank you so much for everything. And we hope to maybe, talk to you again and get some updates in the next, little while if you'd like to come back on and talk to us again. So thank
Sounds
you.
great.
We appreciate it.
Okay,
And thank
take
you to
care.
Vanessa as well, who connected us. So, all right. Have a
Okay,
great rest of your
take care.
evening.
thanks.
Thank you.